All right, so today I'm going to focus on the second part and third part of the cell cycle, which is mitosis and cytokinesis. Keep in mind that the cell goes through three parts: inner phase, which consists of three parts; mitotic phase, which consists of mitosis or the M phase; and cytokinesis, which is the division of the cytoplasm. Mitosis has four parts, and I'm going to talk to you about them now. So the first part of mitosis is prophase. And basically what's happening here through the whole process, the four phases of mitosis, is the division of the nucleus. During prophase, several things take place. First of all, chromosomes coil up. You see here, they basically consist of two sister chromatids. Here's a better picture of what that is. You have a chromatid, and then you got chromosomes or sister chromatids, which is this, or identical chromosomes, which separate. Anytime you see the word chromatid, sister chromatids, they're kind of the same thing. Also, during prophase, the nuclear envelope breaks up. Remember, in interphase, you could see the nucleolus and the nucleus. Here, it's going to be broken up, so you don't really see that part of the cell. Centrioles move apart. Remember, in interphase they were together, and in between them the spindle starts to form. Here is the description of the spindle right here. Let me move this back over. These dots in the center are centromeres, as shown here. You have to know how to label all these parts. Centromere holds the sister chromatids together. Centrioles look like the L-shape or T-shape structure. Your spindle are forming in between them, and the nuclear envelope breaks apart. Second phase is metaphase. Think of the word M for middle, because your chromosomes are going to line up along the equator, which is the middle of the cell, as you see here, and the spindle is going to connect to the structure in the center, which is the centromere. So here you got your spindle, which is made up of microtubules, as you learned in the last chapter, and they connect to the centromere, which holds the sister chromatids together. Anaphase, think of the letter A for away or apart. Basically here, the chromosomes or chromatids move to opposite ends of the pole. This is a picture of anaphase in its initial state, and here's a better picture, anaphase probably in its final stage when they're fully apart. Telophase, which is the last part of mitosis or mitotic phase, the nucleus reforms, as you see here. You could see the nucleolus, spindle disappears. You see your centriole back at the top. Chromosomes are uncoiling. And basically it looks like interphase. The difference is you've got this formation during telophase, which is the cleavage furrow right here. So you still see a cell that looks like interphase, except you see that break within the cytoplasm, and it shows two cells together. The last part of the cell cycle is cytokinesis. Think of C for the splitting of the cytoplasm. And basically you're going to have two cells here that look exactly like interphase. You have your centriole in one end. You could see the nucleolus. You could see the nucleus. The chromosomes are visible, and the envelope is visible. The difference is you're going to see two cells side by side in a diagram, and that shows that cytokinesis took place. Immediately, this cell will begin the cell cycle again, which will go through interphase, and this cell will do the same. Mitosis results in two cells that are identical.